This is a video about COVID vaccine because, like it or not, your decision to take or not take the vaccine can have immense implications on your life and the life of your loved ones. This will not be a cheerleading session for or against the vaccine. We've been given a choice and the vaccines are not mandatory. But though freedom is good, it also invests you with the responsibility to do the right thing, whatever that may be. And to make the decision prudently and responsibly, information is needed. A COVID pandemic has changed our world, humanity has come up with a solution, and now we all need to understand what that is. Let's get to it. We've all heard about antibodies, but to understand vaccines, few more players need to be introduced. This here is a dendritic cell. I know it looks like an alien, but trust me, we are full of them. Dendritic cells are key in fighting off infections. They use their tentacles to sample our bodies for invaders, and when they find one, they break them down into pieces and present them on a proverbial platter for the immune system to recognize. The pieces of the intruder are called antigens, and the dendritic cells are what we call antigen-presenting cells, or APCs. When the antigen is thus shown, it becomes labeled as foreign and the rest of the system kicks into gear. Helper T cells stimulate their B cell brethren to produce antibodies, which in turn act like very specific tags to cover the intruders and single them for destructions. The antibodies rev everything up, and the pathogens get either attacked and killed by natural killer cells or literally devoured by phagocytes. The whole idea of vaccination is to get ahead of the game, to have the antibody tags waiting and kill the virus when it first enters the body, not when it had time to multiply. And it is to the world of the virus where our story takes us next. Viruses are weird. As you know, they are not even alive. If we and our cells are hardware, viruses are nothing but software, packets of pure information that insert their programming into our cells for one purpose: to make more copies of themselves. By now, we are very familiar with this picture representing the virus responsible for COVID-19. The virus is covered with spikes that allow the viral entry into the cell, and since these will play an important role in our story, just remember them for now. But how does the virus turn cells into zombies? Well, let us start with the fact that a cell is a very busy place. It is filled with transportation networks, mitochondrial energy power plants, and actual protein factories called ribosomes. Proteins are what we are made of. They provide structure to our bodies and tiny microscopic mechanisms to infuse us with life. No one knows how many types of proteins exist, but we do know how they are made. As you know, our genetic material is stored in a secure part of the cell called the nucleus, where the DNA resides. Think of DNA as the original blueprint of the body, written in stone, if you will. DNA does not leave its secure location, but instead gets transcribed into flimsy, call it paper, copies of the same instructions, which then float out of the nucleus and instruct the ribosomes to make proteins. There are many types of RNA out there, but one of the most important and most directly responsible for the making of protein is messenger RNA, or mRNA. So how does a virus succeed in taking over the cell? SARS-CoV-2 is an RNA virus. It is a strand of 30,000 genetic letters that flows into the cell, takes over the ribosomes, and codes for 4 proteins that make out its shell and 16 other non-structural proteins that spread its evil. At its core, the main idea of vaccination is to make the dendritic cells present an antigen on its surface. You do that, and the immune system does the rest. But how do we get the antigen to the right spot? Over the years, we came up with a progressively efficient solution to do just that. First, we took small amounts of the real thing, like smallpox, and inoculated a child's arm with it. The immune system conquered the low-dose infection and learned the antigen along the way. Then, we learned to kill some of the viruses and inject their dead bodies in, like we did with polio. But that requires chemicals to kill the virus, and their mangled structures do not elicit good responses in most viruses. Next, we learned to attenuate or weaken live viruses, mutate them just enough to retain their antigens, but to lose their ability to replicate in humans. This is a cumbersome process requiring the virus to pass through hundreds of generations of cell cultures, like eggs. They make it mutate to like chickens and lose ability to infect humans. There's a delicate immune balance there, and people with immunosuppression can get sick from it. The next big step was the recombinant vaccine, a way to engineer the protein antigens themselves and inject those for our antigen-presenting cells to find. So, are there not other COVID vaccines that do not have mRNA in them? There are literally dozens of vaccine candidates out there, but there is a reason why mRNA got here first. The mRNA platform is the most flexible and efficient. No need to grow anything in eggs. It is a simpler, purer process. The next possible candidate is the AstraZeneca vaccine, but I figure if you have reservations about mRNA strands, you might balk at the chimpanzee virus that it is based on, with only 70% efficacy to boot. You know the old saying, give a man a fish, he will eat for the day, teach him how to fish, and he will eat for a lifetime? Well, providing the ready-made antigen to the body is like giving the dendritic cell the proverbial fish. It will eat it, process it, and be done with it. It works for some viruses, but not for others. The dendritic cells fighting the coronavirus need to be taught how to fish, to actually learn from the inside out what the coronavirus looks like. This is where the mRNA technology comes in. The concept behind mRNA vaccination is simple. Identify a suitable antigen protein on the virus, decode its structure, make the RNA template for it, deliver it to the arm, and the wonders of biology will do the rest. The mRNA will float inside the dendritic cells, bump into the ribosomes in the cytosol, and manufacture the desired product. The antigen will then naturally float to the surface and present itself to the rest of the system for the antibodies to be made. Sounds great on paper, but like everything else in life, the devil is in the details, and it is the understanding of those details that will either make you trust or distrust the whole process. So bear with me for a short while longer as we delve into what is shaping up to be the most important story of the century so far. It is crucial to understand that mRNA vaccine technology is not new. Yes, it has never been used outside of studies before, but it has been around for about 10 years, and both Moderna and the Pfizer partner BioNTech have made it a cornerstone of their business since 2010. The story of how we got here is the stuff of legends. The idea of using mRNA to control protein production was suggested eons ago in 1965. It was purely theoretical then because there was no way to get the mRNA inside of the cell. The body views the free-floating mRNA as a threat and destroys it on contact, so how do we go from a needle in the arm to the ribosome inside the cell? There were two main obstacles to overcome. First was to wrap the naked mRNA particle in a lipid pocket, called a lipid nanoparticle, so it can be left alone in the bloodstream and be fused with the cell membrane later. This technology was developed in the 90s and improved on since then. The second issue was the fact that the cells really hated human-made RNA. Even when delivered into the cell, the mRNA would be attacked and destroyed. This was the reason the whole field of mRNA medicine had languished, until this woman, Katalin Karikó and colleagues, figured out the problem. It turns out human cells really hated mint-condition lab-made RNA. Like a savvy car racer who abhors stock but who loves a modified ride, human cells modified their RNA with a natural process of methylation. For years, the body rejected scientific efforts to feed it stock RNA. Karikó discovered the natural way of the human cell. Modify the RNA and the cell will no longer fight it. This was the key discovery that indirectly led to the starting of Moderna and Karikó's senior position at BioNTech, where the Pfizer vaccine gets made. I believe Karikó, whose RNA dreams were rejected time and again, and whose story sounds like a sequel to Queen's Gambit, will win a Nobel Prize for this work. In the meantime, this tissue explains the term modified RNA that has been giving pause to some people. By modifying the stock structure of the molecule, we are simply doing what the body does on its own. The discovery of this natural process is the only reason we are talking about the vaccine at all. Both Moderna and Pfizer vaccines share similar, though slightly different approaches, which include three main categories: genetic mRNA material, lipid nanoparticle pocket, or an oil bubble to keep mRNA protected while in transit from the needle to cell, and inactive ingredients such as phosphate and sucrose, not much different from those found in the Big Mac. In the interest of time, I will focus on the Pfizer vaccine, but Moderna's is very similar. Both Moderna and Pfizer chose a specific part of a spike protein called the S protein. As you can see, even the S protein itself is quite complicated, and the two vaccines are honing in on slightly different parts of the structure. This is important because, as you have heard, there are new virus variants coming out of UK and South Africa, and they both affect the shape of the spike protein. Pfizer and Moderna feel that these mutations do not affect the current vaccine, so far so good, though of course future mutations may not be so kind. So here we are, coming to the end of our story with two salient points remaining. Does the vaccine work, and is it safe? The answers, respectively, are yes, and as far as we can tell. You must have heard on the news that both Moderna and Pfizer vaccines are at least 95% effective. The flu vaccine of 2019 was 45% effective, so these results are fantastic. Here's a graph from the main report showing the rate of COVID infection in those in the vaccine versus those with the placebo. As you can see, it was not even close. No matter what else we can say about it, the vaccine works. But is it safe? This is the main question in everyone's mind, so let's address it head on. There are three sensible issues having to do with safety: the question whether the vaccine research was rushed, the safety of the mRNA itself, and the potential toxicity of the lipid nanoparticles. It really seems that the mRNA technology came out of nowhere and that the vaccine process was rushed. But, as we have seen already, scientists have been laying groundwork for this for decades and started to commercialize it since 2010. Initially, the technology was being targeted at cancer, but the first SARS and Zika outbreaks made people work on vaccines as well. The work was slow until the urgency of the pandemic hit and the science took a leap forward. The building blocks were already here, they just needed to be put together. As far as the approval goes, it was accelerated, but no important corners were cut. In fact, we can argue that all approvals from now on should emulate what was done here. Typical approval process takes years. In phase 1, the vaccine is given to a small number of healthy people to see if it works. Phase 2 studies include hundreds of people where various dosages are tested to see which one is the best. Phase 3 administers to thousands to confirm efficacy and look for rare side effects. Manufacturing is expensive, and it only starts when the data is complete. Here you can see what happened in this case. All the same phases were performed, but overlapped somewhat to save time. Most importantly, the third phase was not rushed, but completed its mandatory 2-month period, which is a time frame in which most vaccine side effects occur. Also, the companies took a bold gamble and started to manufacture even before the approval to have the vaccine ready as soon as possible. Urgency of the matter allowed for fast enrollment, and the studies were completed quickly. This was a feat of organization, and I personally feel comfortable with the process. Second issue bothering people is the injectable mRNA. To review, a strand of synthetic RNA has no chance to integrate into our genes. But do you know who does? The virus. Through its complex machinery, the SARS-CoV-2 virus has already been shown to incorporate its RNA into a patient's genome via a reverse transcription process, so if the purity of DNA you're after, the vaccine is actually the safer way to go. The question that bothered me from the beginning was, how long does the vaccine mRNA stay in people? I mean, we do not want this thing to hang around forever, do we? The answer to this question was surprisingly elusive to find. Neither Pfizer nor Moderna shared that information, and it took me days to track this down. For context, it is important to understand that a cell is totally crowded with natural mRNA all the time. This is not a case of a single molecule waltzing into an empty hole. Rather, this is a crowd of molecules pushing into a crowded station. Our cells excel at cutting up mRNA all the time, and in fact, majority of natural mRNA has a half-time of about 10 hours. In this cool paper, scientists looked at thousands of decay rates of human RNA and found that mRNA degradation varies, with the shortest time given to the dynamic gene expression and the longest to biosynthesis and metabolism. Further work has shown that the rate of breakdown is dependent on the body's own tweaking of what is called the 5' cap structure of the mRNA, a part of the modification system we talked about before. And here we come to the crux of the matter. Though I could not find official mRNA half-times from the manufacturers, I found this paper by the BioNTech CEO Ugur Sahin delineating possible cap modifications and their effect on half-life of the molecule. Two figures are important here. This one shows that various modifications cause different degradation patterns in both immature and mature dendritic cells. Without a modified cap, mRNA has no chance. With caps, it can last longer. How much longer is shown in this table. We do not know which of the caps was chosen to be the vaccine, but here are the choices. The longest half-life here is 27 hours, but you want a cap that not only lasts the longest, but also is most efficient at making protein. This one is particularly poor at protein making, so it was likely out. The best candidate is here. It made the most protein and had a good balance between efficiency and lasting power. So, if I had to guess, I would say Pfizer mRNA levels peak at about 24 hours and dissipate by 60 hours, or about 3 days after the shot. This corresponds well with arm soreness and other effects that often occur the next day and disappear by day 3. The important takeaway here is that no matter what cap, the mRNA is degraded to zero in the end. Nothing lasts forever in a cell, and certainly not mRNA. Third issue is the potential toxicity of the lipid nanoparticles. To recall, these are tiny droplets of fat constructed to keep the mRNA safe in the passage to the cell. They are called nanoparticles because of their small size and have nothing to do with nanorobots or microchips that took over wild imaginations of the internet fringe. Some people worry about the toxicity of these nanoparticles, and that is not entirely without reason. Special attention has been paid to the fact that these particles are pegylated or covered with polyethylene glycol, a substance found in food additives but also used to help the particles survive their journey through the blood. I was personally concerned about the effects of these particles, and this is what I found. The main lipid components of the Pfizer vaccine are these, and there is good basic science study looking at their effects in detail. The study was conducted in rats who were injected with the compounds, and it has served the UK authorities to approve the vaccine. The findings were the following. After injection, the half-life, or the time to eliminate half the substance, of the compounds in the blood was 3 days, while the half-life in the liver was 8 days. It takes about 4 half-lives to eliminate the drug completely, so we can say that 1 month after the vaccination, the particles are out of the system. So there you have it, the sensible concerns about the vaccine. But here is the main takeaway. What matters in the end is not studies in rats or theoretical musings. What matters is data, and so far, data is good. Sore arms is the most common side effect. Some people, especially those with a history of the prior coronavirus infection, get chills, muscle aches, and headaches, but these go away quickly as well. The 2-month follow-up of these phase 3 trials for both Moderna and Pfizer did not reveal any trouble in the time frame. We all wish to know more, and the FDA is honest about lack of the long-term data, but so far, things are encouraging. And here we come to the main point. No one, not the FDA, not Moderna or Pfizer, can guarantee you the safety of these vaccines long term. A year from now, we will know so much more about them. But lack of the guarantee does not mean not to take it. After all, what other guarantees do we have in life? Ideally, we would not have COVID and you would not be watching this tedious video. But here we are with the raging pandemic and new, more infectious variants on their way. So this should not be a conversation about guarantees, but rather of risks and benefits. On one hand, we have the risk of COVID infection, where our cells get carpet bombed by the viral RNA and organs assaulted by severe inflammation. On the other, we have a theoretical risk of a vaccine that has so far done well by over 30,000 people in clinical studies and hundreds of thousands more by the day. Yes, I would love to wait for the scientific papers of 2022, but I do not think we have this kind of time. Even with the precautions we have taken so far in the hospitals and beyond, the new UK variant is about 70% more infective and can be a game changer for us all. So, the decision lies with us now. I hope I've been helpful in making the issues more clear. The decision to vaccinate is personal, and it needs to stay that way. But do separate sinister misinformation from sensible concerns, and, in the end, make the decision on those grounds. No one likes the uncertainty of a novel shot in the arm, but COVID is lurking, and getting avoidably sick would be a real shame. Take care and use the comment section to ask further questions. I will do my best to answer them all. Thank you.